Our bodies are incredible machines, made up of trillions of cells. Instructions are encoded within our DNA. DNA is the blueprint of life. It contains all the genetic information that makes us who we are. Epigenetics has transformed our understanding of genes. It studies how behaviors and environment affect genes. Epigenetic changes are reversible. The human genome is like a vast library. Epigenetics acts as a librarian, determining gene expression. Genes are sections of DNA that provide instructions for building proteins. Proteins are the workhorses of the cell, carrying out a wide range of functions. They are essential for the structure, function, and regulation of our tissues and organs. The process of using DNA to make proteins is a fundamental process called gene expression. Gene expression is a tightly regulated process that allows cells to control which proteins are produced at any given time. This regulation is crucial for normal cell function, development, and response to environmental cues. Gene expression begins with a process called transcription. During transcription, the DNA sequence of a gene is copied into a messenger RNA, mRNA molecule. This mRNA molecule then carries the genetic information from the nucleus, where the DNA is stored, to the cytoplasm, where proteins are synthesized. Epigenetics adds another layer of complexity to our understanding of gene regulation. It refers to changes in gene expression that occur without alterations to the underlying DNA sequence. Imagine a musical score. The notes themselves represent our genes, while epigenetic marks act as musical notations, influencing how those notes are played. These epigenetic changes can be influenced by a variety of factors. These factors include environmental exposures, lifestyle choices, and even our emotional experiences. Epigenetic modifications can switch genes on or off, thereby influencing the production of proteins and ultimately affecting cellular function. There are three primary mechanisms of epigenetic regulation: DNA methylation, histone modification, and non-coding RNA regulation. These mechanisms work together in a complex interplay to fine-tune gene expression. They orchestrate a wide range of biological processes. DNA methylation is one of the most well-studied epigenetic mechanisms. It involves the attachment of a methyl group to a cytosine base in the DNA sequence. Think of it as a molecular switch that can turn genes on or off. Generally, methylation of a gene's promoter region, the control center for gene expression, is associated with gene silencing. This means that the gene is less likely to be transcribed into mRNA. Conversely, demethylation, the removal of a methyl group, can lead to gene activation. DNA methylation plays a crucial role in many cellular processes. These processes include embryonic development, X chromosome inactivation, and genomic imprinting, where genes are expressed in a parent of origin specific manner. Dysregulation of DNA methylation patterns has been linked to various diseases, including cancer. Section 5. Histone modification, the choreography of DNA packaging. Our DNA is not simply a long linear molecule floating around in the cell nucleus. It is tightly packaged and organized into a complex structure called chromatin. This packaging is facilitated by proteins called histones, Histones act like spools around which DNA is wound. This allows for the efficient storage of our genetic material within the confined space of the nucleus. However, this packaging also affects the accessibility of genes to the transcriptional machinery. Histone modifications, such as acetylation, methylation, phosphorylation, and ubiquitination, can alter the structure of chromatin. These modifications can either enhance or repress gene expression depending on the specific modification and its location on the histone tail. Section 6. Non-coding RNAs, the silent regulators. For many years, scientists believed that only a small fraction of our genome, the protein-coding genes, was functional. However, the advent of genomics has revealed that a vast majority of our DNA is transcribed into RNA molecules that do not code for proteins. These non-coding RNAs, or ncRNAs, play diverse regulatory roles in gene expression. They can interact with DNA, RNA, and proteins, influencing various steps in the gene expression pathway. One class of ncRNAs, called microRNAs, or miRNAs, can bind to complementary sequences on mRNA molecules, leading to their degradation or translational repression. Another class, long non-coding RNAs, or LNCRNAs, can act as scaffolds, bringing together different protein complexes involved in gene regulation. These ncRNAs add another layer of complexity to the epigenetic landscape. They highlight the intricate network of interactions that govern gene expression. Section 7. Epigenetics and health. A complex interplay. Epigenetics has profound implications for our understanding of human health and disease. Epigenetic changes are involved in normal development and cellular processes. Dysregulation of these processes can contribute to a wide range of diseases. For instance, alterations in DNA methylation patterns are a hallmark of cancer. These alterations can lead to the silencing of tumor suppressor genes or the activation of oncogenes, promoting uncontrolled cell growth and tumor development. Epigenetic modifications also play a role in neurological disorders, such as Alzheimer's and Parkinson's diseases. These disorders are characterized by the accumulation of misfolded proteins and neuronal cell death. Epigenetic changes can influence the expression of genes involved in protein folding, clearance, and neuronal survival. Moreover, epigenetic changes accumulate over our lifetime. These changes contribute to the aging process. As we age, our epigenome becomes progressively altered, leading to changes in gene expression and cellular function. These age-related epigenetic changes can increase susceptibility to various age-related diseases. Section 8. Environmental influence, shaping our epigenome. The dynamic interplay between our genes and the environment shapes our epigenome. Experiences and exposures throughout life constantly shape our epigenome. Diet, stress, toxins, and exercise influence epigenetic marks. These changes can affect our health, for better or worse. Maternal care has lasting effects on the epigenome. Toxins like air pollution can alter DNA methylation. These changes can increase disease risk. Understanding this helps in disease prevention and treatment. Healthy choices can shape our epigenome for well-being. Section 9. The future of epigenetics. From personalized medicine to evolutionary biology, the field of epigenetics is rapidly evolving. It holds immense promise for revolutionizing our understanding of human health and disease. One promising area is personalized medicine. By analyzing an individual's epigenome, healthcare professionals could tailor treatment strategies. Beyond medicine, epigenetics impacts evolutionary biology. Epigenetic changes can occur rapidly in response to environmental changes.